Welcome to Classic, an ETH 2020 development project that aims to revolutionize the chemical industry by eliminating the need for many intermediates in the synthesis of compounds, thus resulting in a truly sustainable green chemical factory of the future. It will have a strong impact in different sectors, including the production of pharmaceutical drugs and also of fine chemicals.
For this purpose, Universidad Autónoma de Madrid, UAM, coordinates an international consortium of top research groups that will develop a radically new approach to chemical synthesis, inspired by the way in which living systems manage to modulate catalytic activity. A key issue in this direction is being able to control the activity of different catalytic species, making use of the self-assembly and replication of minimal nucleobase sequences. We also aim at regulating the activation-deactivation of those self-assembly and replication functions through external stimuli, for example with light. Importantly, by using the internal feedback mechanisms of the resulting replication networks, we will be able to program their activity at our convenience. Our collaborations with the Gurion University of the Negev, VUU, to implement nucleic acid peptide replicators, and with ETH Zurich to provide them with catalytic properties are critical at this point.
Nature uses replication as the general mechanism for inheritance. One can observe replication across scales, from DNA to cells to human. We aim to mimic this phenomenon using simple synthetic compounds that promote information. We found that short protein molecules contemplate the aggregation and reaction of shorter molecules to form more copies of themselves.
So, we continue to the next level and study the self-organization and self-replication of peptide nucleic acid catalyse. Combining these two types of molecules, we can benefit from both worlds. The peptide drive the self-assembly and the nucleic acid control their hybridization.
Moreover, combining our results and observations with knowledge from the UAM team on the self-assembly of nucleobases, we observed for the first time that in replication networks containing two nucleopeptides, one product replicates much faster than the other, with the molecules featuring survival of the fittest-like behavior. We can now carry out reaction processes to mimic a cell in continuously state-tagged reactor, where constant supply of material and removal of products and waste are maintained. So our next step will be to endow these hybrid peptide nucleic acid replicators with a catalytic potential that will allow them to be used in biocatalytic applications like those carried out by UG and ETH.
At the University of Graz, we are investigating cells' natural catalysts, enzymes. Enzymes enable virtually all processes in living organisms to occur in a defined order and with high precision, which makes them perfect candidates for the creation of cell-like behavior on macroscopic scale.
We are now able to modify enzymes for functions they were not originally evolved for, broadening their scope of application.
Involving light in enzyme-mediated processes allows light-dependent enzymes to form reactions otherwise not observed. Also, light can be seen as a traceless reagent and a sophisticated way of introducing energy to a reaction, enabling it to take place and to generate products.
In the Classic project, we are investigating an enzyme that uses blue light for fatty acids and biofuels and also for other applications. Our aim is to identify a procedure to combine this enzyme with other biocatalysts to make drug-like molecules for renewable research.
We are also looking into the combination of enzymes and short amino acid chains for the same purpose. The compatibility of these two catalysts may also allow us to combine them into a multi-purpose single catalyst. We are carrying out this work in collaboration with the Venomer's group at ETH Zurich.
Welcome, herzlich willkommen at ETH Zurich. I'm Hilma Venemers, and I'm happy to give you some insights what my lab is doing in the Classic project. Nature has evolved enzymes as catalysts. My lab has had a long-standing interest in the question whether peptides that consist of the same building blocks as enzymes but are much smaller can also serve as catalysts. Over the past decade, we have established tripeptide catalysts that are very effective, very reactive and stereoselective catalysts for carbon-carbon bond formations. Now, this is important since it allows for the effective synthesis of organic molecules, and it also raises the intriguing question of the role of peptides in the evolution of enzymes. In CLASI, we combine, together with our partners, peptide catalysis with enzyme catalysis and create multi-catalytic systems. This approach streamlines organic synthesis and addresses the fundamental question whether peptide catalysts are compatible with enzymes. Again, this has far-reaching implications for the production of complex organic molecules, many with therapeutic relevance, and the origin of life.
Radboud University's role in CLASI is to demonstrate how can we perform cell-free biosynthesis in flow while having control of enzymatic reactions.
To make the synthesis of useful molecules cheaper and greener, we need to reuse the catalyst. We achieve that by producing polyacrylamide gel beads using a microfluidics technique. These beads are then used to immobilize enzymes. During the mobilization procedure, the enzymes are attached to the gel beads while lysing residues of proteins. Later, we load them to the reaction vessels, where the chemical synthesis will take place and from which they cannot escape. This means that the enzyme beads can be reused multiple times within the system. By reusing the enzymes, compartmentalizing the reactions, and controlling them, we can perform cascade reactions. This means that the product of the first reaction becomes the substrate of the second reaction. Photoinhibitors will let us choose which reactions and when we want to activate and which one to inhibit in order to synthesize the desired molecules. We can progress on this line of work thanks to a collaboration with Micronit, who provide us with microfluidic devices developed according to our experimental specifications. As soon as the product specification and requirements have been established, CAD software is used to lay out the design of the parts. The microreactor consists of a lower part, a silicone rubber gasket, and an upper part. The lower and upper parts are constructed from multiple layers that are fused together and include a network of microfluidic channels and the filter membrane to retain the catalyst particles inside of the wells. Tubing is connected to the device and allows the user to configure the order in which the reactants flow through the individual wells. Prototypes are manufactured by CNC machining plastic sheets. The machined parts are subsequently assembled and shipped to the CLASI partners for functional testing.